Thank you for the introduction, and thanks also for the opportunity to speak today. I have no conflicts of interest to disclose. So I'll be talking to you today about how to analyze the featureless signal that we get from PGSC that Dr. Nelson just described, because this is what people usually acquire, and it's still nonetheless important to try to extract the most information out of it. So diffusion MRI is interesting because the diffusion length is roughly on the same order of magnitude as the typical microstructural features that we're after. So diffusion MRI is really an excellent probe of tissue microstructure. However, the problem is indeed this very featureless signal here, and in that it's a very indirect probe of tissue microstructure. And then we have to put in a lot of effort into analyzing this signal. So I will be discussing roughly two avenues for analyzing the signal. The first one is signal representations, also referred to as statistical models, and the other one is biophysical tissue models. So roughly, signal representations are formulas to just describe the signal decay without making any assumption about the underlying tissue. Therefore, they are applicable to any type of tissue, which is a pro, and they are also able to provide sensitive biomarkers to processes such as development, aging, or pathology. However, their downside is that they lack specificity and that it's difficult to make inferences about the actual microstructural changes that are going on. On the other hand, you have these biophysical tissue models that are essentially simplified pictures of the medium in which water molecules are diffusing. So here, because you're drawing this picture, these models are each tissue specific, so you have to be careful which one you're applying to what. And however, their promise is really to bring specific biomarkers that characterize the microstructure as opposed to these signal representation issued by biomarkers. And the problem with these models today, but we're working on solving those, is the validity of the assumptions that we're making when we're drawing these simplified pictures here, and also of the fit stability when doing nonlinear fitting of this model signal equation to the actual data. So I'll be discussing first signal representations, then I'll be presenting some biophysical models, really with a zoom on brain tissue models. I don't have time to cover models of other tissues today. And the last part will be devoted to the validation of microstructural features, meaning how can we gain confidence in the parameters that are estimated with these biophysical models. So let's start with signal representations. So of course, by far, the most popular is the cumulant expansion. It's essentially a Taylor series of the log of your signal in powers of BD. So of course, if you stop at the first order in BD, this is DTI here. So you're approximating your signal with the linear function. Of course, you have to stop fairly low in B value for this approximation here to hold. And the minimal data that you need to get your diffusion tensor is 1B0 and then 6 directions on one shell, typically around B1000, as I've mentioned. Now, if you push this one order further, this is now DKI, or diffusion kurtosis imaging. So now, of course, you're approximating the signal with a second order polynomial. You can go up to maybe a bit higher B values, typically B2000 or B2500. And here you're also trying to, starting to estimate how your signal is deviating from a Gaussian medium kind of behavior. So this is also where you're gaining information about your tissue heterogeneity. And in order to extract this information, you need another 15 directions on a second shell, typically around B2000 or 2500. And then you can reconstruct typical maps like mean, axial, and radial kurtosis. Now, something that you have to be aware of is that the series here does have a finite radius of convergence, so you should stop at a B value in, for example, brain tissue about 2,500. Beyond that, actually, the series here, no matter how many terms you add to it, will actually diverge and will no longer approximate your signal properly. So this is also something that you should bear in mind and not try to do DTI or DKI analysis with a B values of, I don't know, 4,000 or so. So one piece of good news is that DKI is really picking up in the clinic. First of all, it's actually really useful because it's been shown to be really sensitive to tissue complexity because you are characterizing how your tissue is different from a Gaussian medium, and therefore it does provide complementary information to DTI in a certain, in a wide variety actually of conditions. And it's also becoming more and more practical. So your standard acquisition, because of course you never kind of go with the minimal acquisition, but the standard acquisition is about 1B0 and 30 directions on 2 shells here. But this now you can probably do within 5 minutes using most recent acceleration options on clinical scanners. And also now we have the option of using a reduced acquisition with just 1B0 and 9 directions, well, 9 specific directions on 2 shells here, whereby you can extract your mean kurtosis. So you cannot extract the full kurtosis tensor, obviously, but you can extract the mean kurtosis. And also you can estimate axial and radial kurtosis if you assume your tensors are axially symmetric. So this is work by Hansen et al., and it's really bringing down the acquisition time for DKI, which will make it more and more used in the clinic. Other signal representations, of course, you can pretty much use any basis of functions that you like to approximate your signal. So here, for example, Özarslan et al. Proposed using harmonic oscillator basis to describe the signal. This is dubbed map MRI. There are other approaches where you consider, for example, that your tissue is constituted of a distribution of diffusion coefficients, and this is where your kurtosis essentially would come from. And then if you assume some kind of function for your distribution here, for example, a Gaussian, you can estimate the mean diffusivity and also the standard deviation of the diffusion coefficients underlying your medium. And you can also just kind of lump it all into one expression here that is referred to as the stretched exponential. So there's actually kind of no physical foundation behind this this formula here. It's really just mathematical, but it really has a lot of applications to tumor characterization, this stretching coefficient here, or exponent. And well, you can go even more sophisticated and now also throw in some anisotropy, so you can consider that your tissue is constituted of a distribution of diffusion tensors now. So this is the diamond model by Scherer and colleagues, where they assume, for instance, a multivariate gamma distribution for this distribution of tensors, and you can estimate an average diffusivity tensor and also the heterogeneity kappa underlying your tissue. And diamond is actually a mixture of well, statistical modeling and biophysical modeling, because they're actually even doing multiple compartments. Within each compartment, you have this distribution of diffusion tensors, so it's getting extremely sophisticated. But there is a talk tomorrow in the microstructure session using this model to a mouse model of TBI, I believe. So you can check that out if you're interested. Now, the main problem with these signal representations, as I've mentioned, is the lack of specificity. So, for example, if you measure in your white matter, for example, an increase in radial diffusivity, what does this mean? This could be the result of demyelination, axonal loss, edema, et cetera, Here I haven't even listed all the possible phenomena that could lead to an increase in your radial diffusivity. And while you're sensitive to a change actually going on, you're unable to tell how the microstructure is specifically changing. So this was really the motivation behind biophysical modeling, was trying to maybe disentangle between these effects here in your diffusion signal. So now I'll move on to biophysical models, and I will just discuss brain tissue models and the long time limit, because I don't have time to cover everything. But essentially, these are the building blocks of most brain tissue models. So you're considering that your tissue is constituted of multiple compartments, which are typically non-exchanging. You have an intraaxonal compartment, which is a collection of cylinders. And in the long time limit, they become sticks, which means that this diffusivity here is 0. And it's just characterized by a diffusivity along the axon here, dA. And these sticks can have some orientation distribution function of your choice. You can say that they're perfectly parallel. You can describe them with any ODF that you would like or you think appropriate, especially for your tissue. Then you have an extraaxonal compartment, which is typically modeled as Gaussian anisotropic. characterized by two diffusivity, DE perpendicular and DE parallel. And you can throw in a third compartment, which is usually CSF, and is modeled as Gaussian isotropic, characterized by one diffusivity here. And of course, each of these is weighted by a fraction. Hopefully, they all add up to 1. And you can see here that in these parameters already, we can try to see how they could be more specific to one type of microstructural change or the other. So typically, the intraxonal fraction here can be sensitive, most specific to a change in axon, so axonal loss. The ODF could be a signature of pruning if you're looking, for example, at brain development in early childhood. A change in DE perpendicular here could be a signature of demyelination. DE par of inflammation, D axonal of axonal injury, of course, and this isotropic, the isotropic fraction, not the diffusivity, of edema or atrophy and so on. So hopefully you can try to say something a bit more specific about the microstructure than you can with metrics derived from signal representations. Now, I don't have time to cover all the models. Of course, you can mix and match these compartments and ODFs however you like. I will just describe a few that are out there and are kind of representative. So a very popular one is NODI, neurite orientation, dispersion, and density imaging. This model is a 3-compartment model, as I've previously described. And for the stick ODF, they choose a Watson distribution here, which is convenient because it's characterized by a single parameter kappa that you can readily relate to the mean cosine squared of the angle between your axons here and the main orientation of the bundle. So a high value of kappa is a highly aligned bundle. A low value of kappa is highly dispersed. So typically, NODI can be applicable to both white matter and gray matter. And so if you count here all the parameters that go into this signal equation, there are 8 of them. And if you try to do nonlinear fitting of all of this to your data, well, you will essentially run into some trouble, as I will show a bit later on. So the way NODI is implemented to try to circumvent this problem is to add simplifying assumptions in order to stabilize the fit. So the axonal diffusivity is said to be equal to the extrastellar parallel diffusivity and fixed to a given value in adult brain. And the tortuosity model here is used whereby the extrastellar radial diffusivity is directly linked to your intraaxonal fraction. And the isotropic diffusivity is fixed to 3, which is standard for free water at 37 Celsius. But nonetheless, out of 8 independent parameters to estimate, we are left with only 4. And if you look at these 4 parameters here, they only characterize the geometry. So you have the fractions of your compartments and also the orientation dispersion, essentially. So if you have any change here in diffusivities, this will spill over as a change in these geometry parameters because these are all fixed or tied to another one. So the problem is that in this case, you have a loss of specificity when you're actually looking for specificity when trying to do this biophysical modeling effort. So this is an issue. And another approach for modeling here that I will briefly describe is WMTI, white matter tract integrity. This is a bit different from other models because it involves a direct derivation of model parameters starting from the diffusion kurtosis tensors. So here you can get away with linear fitting, which is nice. But of course, you also have some strong assumptions underlying this model in order for these relationships here to hold. So this is a 2-compartment model. You don't consider CSF. And very importantly, the intraaxonal and extraaxonal spaces are both modeled as Gaussian compartments in order for each of them to be assigned a tensor. So you consider that your collection of sticks is still Gaussian, which is a strong assumption. So you really need to have limited orientation dispersion. Typically, ideally, they should be perfectly parallel. And in practice, you can say that it's still valid under very limited orientation dispersion. So this model is only applicable to very highly aligned white matter bundles. Okay, please don't fall asleep. But of course, this is in the, thank you, in the name of the model, so that's not a surprise, but it is an assumption. And then here you also have this quadratic relationship between the kurtosis and your compartment diffusivities, whereby you need to make a choice of sign here when inverting it to characterize the extracellular and axonal diffusivities respectively. So in this model, it was chosen that D axonal is less than DE parallel, but it could be the other way around. And this is also something that I will spend some time on in the forthcoming slides. So if we try to compare quantitatively the model estimates, you will see that the assumptions actually really impact your, well, your quantification. So this is a study that we've done on development in children from 0 to about 3 years old, looking at white matter tract characteristics, comparing NODI and WMTI. So here you can see that at least the trends with age match qualitatively, but you can see that the quantitative estimation of your fraction, for example, is much higher in NODI than in WMTI, and here the orientation dispersion is less in WMTI than it is in NODI. With NODI, of course, the diffusivities are fixed, and the ones that are estimated in WMTI actually don't match the values that are fixed in NODI at all. So you just have to be a bit careful when actually taking the output of these models for granted, because there are assumptions going into there. And the next thing we wanted to do, therefore, is to try to see if we can kind of alleviate all of these assumptions, because we're not sure how valid they are. And so this is work that we've put into characterizing a two-compartment model that I've dubbed this time NODI-DA. It's a bit of a pun, but so this is now a two-compartment model, so we've got rid of the CSF just to simplify a bit. But this time we try to estimate absolutely all the parameters in the model. We don't fix anything. So we are left with 6 independent parameters to estimate. And what happens here, so even if you take very high quality data, for example, Human Connectome Project, you see that your distribution of parameter values in an ROI that is very homogeneous, for example, the posterior limb of the internal capsule, it could be corpus callosum, it could be anything. You see that you have a bimodal distribution here of values, and even if you take a single voxel and you run the fit, just changing the initialization, you have 2 possible outcomes of solutions within a single voxel. So essentially you have 2 highly probable solutions to your two-compartment model here that represent very different biophysical realities. And essentially, we just refer to them as one being D axonal being less than DE parallel, and the other one, the other way around, D axonal would be higher than DE parallel. So again, you see that you have to make this choice at some point, and we don't know yet which one is correct. And the reason there are 2 possible solutions is that if you look at the fitting landscape of this problem here, you do have 2 completely distinct minima in parameter space. And also around each of these minima, you have an ensemble of very probable, well, low objective function values, so very probable solutions to your model. And with noise, you can travel pretty much anywhere along this pipe and end up with parameter estimations that are very far from your ground truth. And this degeneracy here is actually a very general feature of a two-compartment model, as has recently been shown analytically by Novikov and colleagues. So here now we have a very general model with a scalar kernel characterized by the parameters that I've been describing until now, and a very general fiber ODF here that where we make actually no assumptions about the specific ODF. And you can estimate these model parameters either by using a Taylor expansion if you stop at fairly low B values, or you can push it to with a much higher B values, using nonlinear fitting. But in both cases, you can show analytically that you do have 2 solutions, and you do hope that going to higher B value will kind of solve this degeneracy. And this is something that will be discussed on Thursday afternoon in the power pitch session. I invite you to attend that as well. And the last thing that I want to mention, so we have these 2 solutions, and can we decide which one is correct based on goodness of fit? The answer is no, because here you can see in this axial slice here, I've kind of color-coded the voxels as to which solution would be chosen based on goodness of fit, whether it's dA less than dE parallel or the other way around. And you can see that it's really almost random across the brain. So you cannot rely on goodness of fit to determine biologically which solution is most plausible. So here are 2 points that I think, well, at least to me, are good practice for biophysical modeling. Please be critical of the output. So if the trends that you see are not consistent with common knowledge about disease, aging, etc., it is probably the model that is inappropriate. It's not necessarily that you made some groundbreaking discovery. I mean, you might have, but just double check. And also, don't dismiss signal representations because the sensitivity is there. What I often hear about biophysical models is, well, at least it's multi-shell, it's still better than DTI. And I'm like, yes, so is DKI. So yeah, just if all you care about is sensitivity, do stick with, for example, signal representations. So the last part of the talk now is trying to validate these microstructural features nonetheless that we get from biophysical modeling. And sorry, I just need to catch my breath. And the first thing that is important is to try to validate the stick model for the axons in the long time limit. And this has been done very recently by Verard and colleagues. This is also a presentation tomorrow afternoon in the microstructure session, where essentially at very high B values, your signal should vary as B to the power minus 1/2 if this model is to hold. And this has now been observed experimentally. So this validates a posteriori a lot of models that we have been using for a while, which is good news. And the other thing that we can try to validate is the external volume fractions, try to compare them to estimates from light or electron microscopy. So this here is work by Jespersen et al., where they tried to look at correlations of their fraction estimated in their models with the axon fraction estimated from the electron microscopy. And they had extremely good correlations. However, you also have, for example, very good correlation with your third eigenvalue from DTI. So the fact that you have a very good correlation doesn't necessarily mean that your fractions match 1-to-1. So you also have to be careful about how you interpret those. And this is another work which is very nice by Stickoff et al., where they looked at the macaque corpus callosum, and the external volume fraction from MRI was estimated with NAUTI, and they also compared it to the external volume fraction from electron microscopy, and the quantitative match was actually very good. So this is also encouraging. And this is work by, well, myself and colleagues with a cuprizone mouse model of demyelination, where we've shown that external water fraction estimated from WMTI actually matches very well with the external water fraction from electron microscopy. And also the extracellular radial diffusivity here correlates very well with the G ratio estimated from electron microscopy. And both of these are actually something that you would expect analytically in terms of parameter specificity. However, there are some limitations to validating external volume fractions against light and electron microscopy. First of all, are these really the ground truth? You can have distorted slides. Your fraction can be altered by fixation, so you also have to be careful there. You have a really strong mismatch of spatial resolution between the two methods. And typically, you're sometimes comparing in vivo to ex vivo. So the quantitative match remains very challenging. And again, I want to stress that correlation does not prove specificity or quantitative agreement between the metrics, so you also have to be careful. Now, the other biomarker that we can try to validate is the orientation dispersion of the axons. Now, I don't know of a study actually that compared estimates of orientation dispersion from biophysical modeling to those from other methods that I will briefly mention here, but at least we have now some guidelines as to what to expect in rat corpus callosum and human corpus callosum. So for example here, this was done using myelin staining of rat brain sections and then through slice myelin fiber vector lines. So this is work by Lergaard and colleagues. So they estimated this dispersion in the rat cortex to be about 14 degrees. corpus callosum, rat corpus callosum to be about 14 degrees. So this is, of course, corpus callosum is supposed to be very aligned. Typically, we assume the axons to be perfectly parallel, but you can see already there that we have some dispersion. And this is actually very consistent with the estimate in human corpus callosum using a structure tensor analysis of microscopy images by Budi and Anesi. And also, in another study, we have a very good agreement between the orientation dispersion estimation from the structure tensor analysis and that we get from diffusion of NAA in the human corpus callosum. So at least this gives you some guideline of the orientation dispersion that you can expect in the corpus callosum, so you can compare it to what you get from your biophysical modeling estimate. Now, the big chunk that remains is the validation of compartment diffusivities. As I've mentioned, there's really this question mark here around the inequality, which one is higher, inside or outside. And the problem is that NMR is really the method of reference for measuring self-diffusion coefficient of water. So it's very hard to cross-validate with other methods. And you also cannot infer diffusivity of water from that of other molecules. So now, your best hope is really to try to find some water NMR-based orthogonal measurement that will enable you to tell which one of these is correct. So this kind of merges with, well, some aspects of the previous talk. So for example, here, using isotropic diffusion weighting that was described earlier in the human brain, it's been shown that the isotropic kurtosis here is overall extremely low. So this is synonymous with the fact that the compartment traces are very similar. And this is also corroborated by an earlier study that used gadolinium perfusion to suppress the extracellular signal in the rat brain, where they showed that the diffusion tensor trace before and after perfusion was also unaltered. And this is now consistent with the fact that D axonal would be higher than DE parallel. And other studies here using double diffusion encoding in the rat spinal cord by Skinner and colleagues have shown that the axial diffusivity does not change before and after extracellular water suppression using this method. So they concluded that D axonal was actually similar to DE parallel. And this is work that I will be presenting tomorrow in the microstructure session, where we also suppress the extracellular signal using gadolinium in the rat corpus callosum. And we also have no change in axial diffusivity before and after the perfusion, whereby we concluded that D axonal should be at least equal and maybe higher than DE parallel. So this is kind of where we stand for now. And the last most recent developments are including time dependence or compartment T2s to lift this degeneracy. And I will not spend time on this, but this is work by Olson and colleagues and Verart and colleagues respectively that will both be presented Wednesday afternoon in the time dependence and relaxation session. And both studies also conclude that D axonal should be at least higher than DE parallel. So to sum up, signal representations are very sensitive to tissue microstructure. Their downside is that they lack specificity. But I think it's important, again, to stress that DKI does provide information on complexity compared to DKI, and it's now becoming really clinically feasible. There are other representations out there, and we're really looking forward to hearing from them on clinical data. And regarding biophysical tissue models, in theory, they should provide specific metrics of microstructure, but in practice, they are dependent on the validity of the model assumptions and on the fitting procedure. So please, for now, do interpret the output with caution. And there are great recent validation efforts that have been made, so this is really encouraging. More are welcome. And the open questions that remain are, again, about compartment diffusivity values. And the other one that I didn't really mention, but that is very important, is the cell membrane permeability. And roughly, is myelin necessary to restriction? Meaning, in the gray matter, would we just see myelinated axons in this stick model, or would we actually see all dendrites? And with that, I would like to acknowledge the people that contributed to the work that I presented from our side and funding sources, and thank you for your attention.